You know, we spend billions of dollars developing these incredible cancer drugs, specifically immune checkpoint blockades, things like anti-PD-1.
Right.
And for some patients, let's be honest, they're practically miracles. They just melt tumors away. But for a huge percentage of people, they just fail.
They do.
And for a long time, we thought the failure was, in the tumor itself, or maybe the drug wasn't strong enough. But this new paper we're looking at today suggests the failure might actually be in the intestine.
It's a massive paradigm shift. I mean, we're looking at a study published in Nature by Najjar and colleagues, and it fundamentally changes how we think about immunotherapy.
How so?
They didn't just find that gut bacteria help. That's old news. They mapped a specific causal chain where a single gut bacterium literally trains a special ops team of immune cells, sends them across the body, and orders them to change their identity to fight cancer.
Okay, that sounds like science fiction. But this is hard biology.
Yeah.
The paper is titled Microbiota-Induced T-Cell Plasticity Enables Immune-Mediated Tumor Control. It is. And I want to be really clear right off the bat, we are not just telling you to eat more yogurt here. We're diving deep into a mechanism, antigen mimicry and T-cell plasticity.
Exactly. This is all about the mechanics of the immune system. The nuts and bolts.
So let's set the board. Usually when we talk about immunotherapy, we use that lock and key analogy. The drug is the key that unlocks the immune system's brakes, right?
That's the standard view, yeah.
Yeah.
Anti-PD-1 takes the brakes off the immune system's car. But this paper argues that even if you take the brakes off, the car won't move if the engine isn't running.
And the gut bacteria.
The gut bacteria are what turn the engine on.
Okay, so let's look at the setup in this study. They describe a triad of control. We have the drug, anti-PD-1. We have the bug, in this case, it's a very specific one, Segmented Filamentous Bacteria, or SFB.
Right.
But usually in these highly specific mouse models, you need a target on the tumor too. Did they? Did they have to engineer the cancer to match the bacteria?
You hit the nail on the head. That is the crucial part. They used a melanoma model, B16F10, but they genetically tweaked it. They made the tumor express a specific protein fragment, an antigen, that is naturally found in that SFB bacteria living in the mouse's gut.
So this is the whole concept of antigen mimicry. The tumor has to be wearing the same jersey as the bacteria, or the immune system just doesn't care.
Precisely. Think of it like this. The SFB bacteria in the gut is a wanted poster.
Okay, I like that.
The immune system sees this bacteria in the intestines. It studies its face, the antigen, and it creates T cells that are specifically designed to hunt anything that looks like that poster.
Yeah.
If the tumor doesn't have that face,
the immune system walks right past it.
It completely ignores it. And they proved this wasn't just some general health boost by using a control tumor, right?
They did, yeah. What was that?
They used a control tumor called B16EV that didn't have the antigen. In those mice, having the SFB bacteria made absolute no difference. The drug failed completely. Which proves that the bacteria isn't just boosting the immune system like a vitamin. It is providing specific targeting coordinates.
That is a frightening variable, though. I mean, it implies that the efficacy of a life-saving drug might hinge on whether a patient's tumor just happens to coincidentally look like a bacteria they encountered 5 years ago.
It connects the two worlds completely. Without the bacteria to prime the system, the drug is, in this context, useless.
So, just to recap the setup before I move on, about 5 minutes in, we need the drug, that's the anti-PD-1.
The gas pedal, yep.
We need the SFB bacteria in the gut, that's the trainer. And crucially, the tumor has to be wearing the wanted face, the antigen match.
That is the perfect summary. You need the drug, the bug, and the match.
Okay, but here's where the logic gets tricky for me. The wanted poster is hanging up in the small intestine. The tumor is on the skin or in the lung. I mean, that's a long commute for a cell.
The very long commute.
How do they get there, and who exactly is making the trip?
This is where the paper gets just incredibly detailed. The sheriff in this story starts out as a cell called a TH17 cell.
TH17, okay.
Now, normally, TH17 cells, they just hang out in the small intestine lamina propria, the CLP. Their job is barrier protection. They produce a cytokine called IL-17A.
Right, I remember TH17s. They're usually the ones keeping the gut lining intact, making sure bad bacteria don't leak out. They're basically maintenance workers.
Exactly, they're homebodies. But the scientists used a technique called fate mapping to see what happens to them.
Oh, I love fate mapping. It's essentially like biological tagging, isn't it? Like a little GPS tracker on a cell.
It's the perfect analogy. Imagine dipping a bird's feet in bright neon paint.
Right.
No matter where that bird flies, whether it goes to a tree or telephone wire miles away, you can look at it and say, that one, That one came from the paint bucket.
So they genetically painted these specific TH17 cells in the gut.
They did, so they could spot them anywhere in the body.
Yeah.
And I assume they found them far from home.
They absolutely did. They tracked these specific neon-painted cells leaving the intestines and traveling all the way to the distal tumor site. But, and this is the plot twist, when they looked at the cells inside the tumor, they had changed.
Changed how? They underwent plasticity or, trans-differentiation. Once these TH17 cells entered the tumor microenvironment, they stopped acting like gut barrier cells. They transformed into TH1-like cells.
Wait, so they completely changed their job description?
Completely. In the gut, they're making IL-17A to protect the wall. In the tumor, they stopped making that and started pumping out interferon gamma and tumor necrosis factor.
Whoa, those are warheads. Those are pro-inflammatory attack signals.
Like a construction worker traveling to a war zone and retraining as a demolition expert on the fly. And the fate mapping proved it was the same cell.
It wasn't a new cell replacing the old one.
No, the gut cell evolved.
That challenges a lot of what I thought I knew about cell biology. We usually think of cell identity as, fixed. I'm a liver cell, I'm a T cell, but you're saying the environment dictates the identity.
The environment and the mission. If they stayed as regular TH17 cells, they wouldn't be effective against the cancer. They have to trans-differentiate to release those specific cytokines, IFN gamma and TNF, because those are what remodel the tumor environment.
Okay, so quick recap here. We've tracked the cells from the gut to the tumor. We've established that the origin is SFB training these TH17 cells. They migrate. Then this key step, plasticity, where they become TH1-like cells.
And they switch their weapons from barrier defense to these high-impact cytokines.
So visualizing this demolition expert arriving at the scene, we have the gut-trained cell. It's traveled, it's swapped its tool belt for explosives. Is it doing the killing itself, or is it calling in an airstrike?
That is the crucial distinction. These XTH17 cells are primarily coordinators. Their main job is to use that interferon gamma to make the tumor highly visible. They recruit and activate CD8 plus cytotoxic lymphocytes.
Oh, the CD8s. Those are the real killers.
Those are the heavy hitters. The XTH17 cells essentially paint the target with a laser, so the CD8s know exactly where to strike. They enhance antigen presentation, making the tumor cells wave a giant flag saying, "I'm here, come get me."
But this brings up a massive question. Why do we need the gut cells to do this? The body has plenty of T cells. Why go through this elaborate train in the gut, travel, then transform process? Are these XTH17 cells special in some way?
That's the million-dollar question. And the answer seems to be yes. The researchers found that these XTH17 cells act like super soldiers.
Super soldiers. Okay. That implies they can do something regular T cells can't.
Exactly. In a tumor, you have these pesky cells called Treg's regulatory T cells.
Peacekeepers.
The peacekeepers, or, the wet blanket of the immune system. Their job is to shut down inflammation to prevent autoimmune damage. And tumors love Tregs because they protect the tumor from the immune system's attack.
So they accidentally protect the bad guy?
Right. But these gut-derived XTH17 cells, they seem to have a higher threshold for suppression. Because they were born in the hostile, complex environment of the gut where there are trillions of bacteria, they just don't get turned off as easily as a standard T cell.
Huh. That actually makes intuitive sense. If you learn to fight in the mud and chaos of the microbiome, the tumor environment might not be as intimidating.
Exactly. And when they looked at the ratio of cells inside the tumor, the presence of SFB bacteria drastically increased the ratio of CD8 plus killers relative to the Treg suppressors. It just completely tipped the balance of power.
So let's just pause and look at the whole picture here, because this chain of events is wild.
That's amazing.
The bacteria trains the cell, the TH17. The cell migrates. It changes its identity plasticity to become a demolition expert. And because it's a super soldier from the gut, it ignores the Tregs trying to stop it and calls in the CD8 airstrike.
That is the complete mechanism. And they proved it was necessary with an ablation study.
Right, the ablation. That's where they delete something to see if it breaks the system.
They genetically deleted the specific IL-17A precursor cells, the gut trains. Even with the drug, even with the bacteria present, if you deleted those specific recruits, the tumor just kept growing.
So it's a strict causal chain. You break one link, no gut training, and the whole therapy fails.
Correct.
Okay, but here's the catch, and I feel like there's always a catch with these mouse studies. If I'm a patient or if I'm thinking about clinical application, can I just take this bacteria at any point? Or is the clock ticking?
The clock is definitely ticking. The researchers performed what's called therapeutic gavage.
So basically feeding the mice the bacteria at different times.
Exactly. And they found that if the tumor's been there too long, it's already built its fortress.
The window closes?
Fast. Early gavage, days 8 to 12 after the tumor started, worked brilliantly. But late gavage, days 15 to 19, it had diminishing returns.
Why is that?
It suggests there's a critical window for immune priming. You have to introduce the wanted poster early while the immune system is still assessing the threat. If you wait too long, the tumor environment becomes too suppressed, too many tregs, too many walls, and the super soldiers can't get a foothold.
That is a huge clinical reality check. It means timing isn't just a logistical detail, it's a biological variable. You can't just throw probiotics at a stage 4 cancer and expect a miracle if the window is closed.
Absolutely. But to give a bit of hope on the scope of this, they didn't just stop at skin cancer.
Right, they looked at lung and colon too.
Yes, lung cancer and colon cancer. And the same mechanism applied. As long as there was an antigen match between the bacteria and the tumor, SFB helped control the cancer.
So the mechanism seems to be universal, even if the timing is tricky.
It's a fundamental principle of how the immune system bridges the gut and the rest of the body.
Okay, so let's do our final recap. We've seen that the TH17 cells coordinate the attack, they recruit the CD8 killers, they overcome the tregs.
And we know they're necessary. Without those gut-trained cells, the drug fails.
And timing is everything. Early exposure is crucial. Plus, this isn't just for melanoma. It works across skin, lung, and colon cancer models.
It's a very thorough piece of work.
But let's be real for a second. We are not mice. And SFB, this segmented filamentous bacteria, it isn't a major player in adult human guts, right?
That is the most important caveat. This is proof of principle. We cannot simply infect every cancer patient with SFB. In fact, SFB is rare in humans after childhood.
So we can't just go to the pharmacy and buy an SFB pill to cure cancer.
Not SFB specifically, no, but the concept. That's where the revolution lies. Imagine the future of probiotic vaccines.
Break that down. What would a probiotic vaccine actually look like?
Well, now that we know the mechanism, we don't need to rely on luck. We could potentially engineer a safe, common gut bacteria, like an E. coli or a lactobacillus, and genetically modify it to express the antigens of a patient-specific tumor.
So you're saying, you take a biopsy of my tumor, you find the unique proteins, you put those proteins into a harmless bacteria, and I eat a yogurt containing that bacteria.
Theoretically, yes.
Yeah.
You take this engineered probiotic, your gut would see the antigen, train these super soldier T cells in the safety of your own intestine, and send them out to the tumor.
And then?
Then, when you take the anti-PD-1 drug, your immune system is already primed and hunting. The army is already in position.
That is just mind-blowing. Instead of just poisoning the tumor with chemo or hoping the immune system figures it out, you're essentially programming the body's own logistics network to fight a war on multiple fronts.
It completely redefines the relationship between the gut and the tumor. We used to view them as separate worlds. This paper proves they are connected by a physical highway of immune cells.
The gut-tumor axis isn't just a buzzword, it's a deployment route.
Exactly. And it shows that the solution to complex diseases often lies in understanding the systems we already have, rather than just trying to overwrite them.
This has been an incredible deep dive. To think that a cell trained to protect your intestinal lining could change its clothes, travel to your lungs, and lead an attack on cancer, nature is just absolutely relentless.
It is. And it makes you wonder what else the microbiome is doing that we haven't even mapped yet.
So here's a provocative thought to leave you with. If the gut microbiome is essentially a training camp for our immune system's special forces, what happens when we disrupt that camp with antibiotics or a poor diet? Are we accidentally demobilizing our own army before the war even starts?
That is the question we all need to be asking.
And for you listening, if you could design a bacteria to teach your immune system to hunt a specific disease, not just cancer, what would you target? And does knowing your gut bacteria are training your immune cells change how you think about your next meal?
It certainly changes how I look at my breakfast.
Right. I'm host speaker.
And I'm expert speaker.
And this has been The Deep Dive. Thanks for listening.